So it's this one. So just maximize a bit. So before Dr. Baticalan starts. So this is from the Big Brain Project. So it has been given its own website in the European Union. So this is your brain, and we can actually cut through the brain without slicing someone's brain. So this orientation is coronal, so meaning you're looking at a person face to face. This is eggshell, so it's drag it a bit. So eggshell meaning front is here, occipital is here. This is your convention is this is the right side of your brain. This is the left. So you're looking at the brain from below, and this is the brain coming in from the side. So cutting your brain from ear to ear. So the good thing about this website is you can actually drag it depending on which part you want to see. So before MRI was invented, people only had CT scans. So we'll deal with how a CT scan looks like by looking at how the brain is cut by a CT scanner. So I use this. So we're moving. You'll want to focus your eyes here. So and we were now at the cerebral cortex. So I'm cutting the brain from below, going up. So here you see the temporal lobes, and that's the orbital part of your frontal lobe, as you can see here. And we want to cut going towards the vertex. So this is the part where you see the midbrain. Sorry about that. So as we ascend, so this cut shows you the frontal, the insula, the temporal lobe, occipital lobe down here, and that's a bit of cerebellum down here. Those are the CSF pathways. And if we go higher and higher, we see the internal capsule, the boomerang. So as we proceed, so these chambers here are the lateral ventricles of the CSF pathways. So we keep cutting upward. So there was a person who trolled my YouTube upload about this particular website. He's complaining, why are you using a abnormal brain? So this is an abnormal brain. I pointed it out to class 2025 that this brain is not completely normal because there's a stroke here. So there's a cavity there. That was a small hemorrhage, a bleeding in the brain. Well, it may have been asymptomatic, meaning walang nararamdaman yung patient. Well, anyway, so we're still at the level of the internal capsule, and we keep moving upward. So here, the internal capsule gives way to the corona radiata. So we're just at the ventricles, already went past the subcortical gray matter. So we go up higher, we see the ventricles, and then the ventricles will start to disappear. So let me bring this down for you. So this whole area here is the centrum semiovale. So as we proceed, we bring the brain further and further up until we reach the very vertex of this brain. So we've reached the top. So now let's reset. Now we want to see it coronally. So we'll move the brain. This is the blade over here. So we'll move past here. Focus your attention on this panel over here. You can also look at it here. So I'm going to move the brain towards the blade. So you cut the brain like so. So I always like to ask the students, how do you tell if this is a frontal lobe? You can tell because of the straight gyrus here. I also like to think of it as a triceratops, a pair of triceratops facing each other. So this is frontal lobe. So let's move the blade a bit. So we see the triceratops. So here we see the cingulate gyri facing each other. So we're now here in terms of we are cutting the brain. So we move and move until the ventricles start to appear, and move forward a bit until the The basal ganglia appear. Those are the ventricles. That's your corpus callosum. So as we proceed, we see the basal ganglia, then the anterior limb of the internal capsule appears. Then this is where you see the anterior commissure. So right below that anterior commissure is this important basal nucleus of Maynard. Okay, so we move past the anterior commissure. You see these are the optic nerves coming out from beneath the diencephalon. And then we start to see the brainstem here behind. We're already here. As we proceed, you see the midbrain and the brainstem here, the pons. And then we cut further backwards. That's the genu. That's the splenium of the corpus callosum, very thick. This is the pineal gland over here. So Dr. Sang will lecture to you that on a new lecture. And then as we proceed, we're approaching the back part of the brain. Now, before I miss out on it, going back in front, so at this level, looking at the temporal lobes, if you focus your attention here, there's a gray matter here that's in charge of your fear and your rage, part of the limbic system. That's the amygdala. Then as we go back, it gives way to this curled up figure here inside your parahippocampal gyrus. That's your hippocampus. And the hippocampus extends all the way back until we're actually at the occipital. So sort of connects your memories with what you see and what you hear. You know? So memory and visual memory, for those who think you are visual learners, so that's your hippocampus, which proceeds all the way back to the occipital lobe here. So it disappears now. Just where your splenium appears. Then as you proceed posteriorly, you see now the occipital lobes. Now, how would you differentiate this with the frontal lobe? If you're cutting it, there is no straight gyrus there. So comparing this cut with this cut, so Triceratops versus not Triceratops. Okay. Then if you finally you want to end it with cutting the brain from ear to ear, we'll use this one. You'll use this as the cutter. We'll just move the brain. Okay, we're starting from the left ear. So focus your vision on this frame. We cut, cut, and cut, and cut until you see the brain. So that's temporal, frontal, occipital. You see here the hippocampus and the amygdala in front of it. Then we see the basal ganglia, and now we see the corona radiata giving way to the internal capsule inside here. Until we reach the center of the brain, where there's a corpus callosum, and you're supposed to have a septum, a membrane separating the left from the right brain. Until we reach the right side of the brain, so that's corpus callosum already. Those are the diencephalon in the midline. And then that's the splenium. Okay, comparing the splenium, the body, and the genu over here. And proceeding to the right side of the brain. You see the basal ganglia, and now we see the internal capsule on the right side, which will give rise to the corona radiata here. Those radiating fibers here, which becomes the centrum semiovale up here. Okay, so that ends our pretend brain cutting. And if you have any questions, we have 8 minutes remaining. I'll send these links to your liaison.
There's one question for right now.
Okay.
Which structures in the brain are affected when an individual loses their memory due to alcohol consumption?
Alcohol can cause degeneration in your three-layered parts of the brain. So, namely, your hippocampus and your cerebellum. This can degenerate because of chronic alcoholism. So that's why your memory problems happen, and you have cognitive problems as well.
Tapos, Doc, do you have any tips po on how to remember all of these structures? Kasi medyo info overload.
Yes, these are introduced to you. Well, what we do here is we repeat. No, we keep repeating. So we go layer by layer. So this is superficial. It's just I get to know you with the structures here. And then you will go back to them one by one in the different functional lectures. So initially it's just anatomy. Then we're going to functional anatomy, meaning what they do and putting it all together. At the same time, how to test. No? So we'll be testing different parts of the brain. Don't worry about it because this is the onion peeling scheme, no, of learning in OS202. Some of you might find it repetitive if you don't like repetitiveness, but repetition is key when you want to remember things.
Tapos, ano pa, what is the basis of having a patient awake or asleep during brain surgery?
Well, we'll have a session on that. No, we'll invite Dr. Aguilar, Dr. Legaspi, as well as Dr. Seng and Dr. Baticolon. The reason why we do awake surgery is if we're dealing with very so-called eloquent parts of the brain, meaning the parts of your brain that are in charge of speaking, moving your limbs. The reason why they use awake surgery is the patient is cooperative enough, so they will stimulate that part of the brain they're about to cut and see if they stop talking or if they start acting strangely or if they cannot move their limbs anymore. Because as I mentioned, what you see on the gyri and sulci are not necessarily where your patient's neurons are actually residing. You may more or less know the area, but the actual gyri and sulci, it's not exact. That's why the awake surgeries are very helpful now. You'll hear more about that from Dr. Gap Negaspi.
Okay po. Are the centrum, semi-ovale, corona radiata, and internal capsule all part of the corticospinal tract?
Well, the corticospinal tract is actually one of the many fibers that pass through those steps. No, centrum semiovale. When you're closer to the cortex, closer to the neurons themselves, the corona radiata becomes such because it has something to pass over, which is the ventricles of the cerebrospinal fluid pathway. And then after getting to that barrier, it now has to squeeze figuratively through the eye of the needle, which is between the in between the gray matter in the subcortical areas. So it from big to small.
Tapos some people are asking po about the coverage for the first formative exam.
Yeah, I'll be closer to the date because there's still some upheavals in the schedule because of Dr. Nagaspi's availability. So definitely the first part will be anatomy, mostly anatomy.
Okay po. Yeah, I think there are no more questions.
Okay, it's really overwhelming at the start. So we're just giving it to you one onion peel at a time. Okay. So that's why we're not. Oh, we have a raised hand somewhere. Someone's raising their hand.
Hi,
Yes.
Doc. I wanted to ask with regards to the corpus callosum because I remember in my undergrad we talked about how cutting the corpus callosum can reduce seizures.
Yes.
And then you also mentioned that it's really bad to cut it, but we also knew that parang when you cut it, there were deficiencies in their cognitive capabilities. But like, how realistic or how possible is it to cut it,
Yeah.
the corpus callosum?
In terms of epilepsy surgery, so these people are suffering for most of their lives. They're taking several drugs and they're still having seizures. So the extreme sacrifice, well, now with awake surgery, they can tell, no, okay, if we cut this part, what happens? But extreme cases, they will cut usually the genu if they want to stop. They will not cut the entire corpus callosum. It's like cutting that person into half, no, into two different people. So it's not the whole corpus callosum that will be cut. It's just you're disconnecting the source of the electrical activity from the rest of the brain and hopefully stop the seizures or make it more manageable.
Okay, thank you so much, Doc.
Okay. We have 2 more minutes before Dr. Baticalan starts. Yes, you have another raised hand somewhere.
Hello, sir.
Hi.
Hi, sir. I just wanted to ask, sir, about the sulci, because you mentioned earlier how they're mostly, at least in our discussion, they're mostly for demarcating the gyri. And
Yes.
I was just wondering if the depth of the sulci is actually important, like functionally. Like what would the implications be of like having sulci that's too deep or too shallow?
Well, deeper is better. So we have conditions. Some people are actually born without any wrinkles on their brains, which, as you may surmise, their intelligence is below average. So the deeper the sulca is, that means more neurons formed. That means more folding occurred. And folding originates from where the origin of those gyri are actually deep inside the sulci. So the more important the sulci is, the deeper it is and the more cells. And the main example of this is the Sylvian fissure. As you will learn soon enough, the Sylvian fissure, the brain surrounding the Sylvian fissure are actually the ones that subserve language. So important. And the next important divider is the central sulcus, which demarcates motion versus perception. So wherever the deepest cuts are, that's where the most multiplication of neurons occurred early in life. The more differentiation occurred, and the more it's like for those of you who've taken Earth sciences, it's like the seabed of the floor. No? How islands form because more volcanic activities leads to more islands being pushed out of the Pacific Ocean. The same concept applies during brain development.
Sir, so with that, sir, I was wondering, so is there such thing as a sulci being too deep?
There are sulci which are too deep, which are actually abnormal already. So they are called cysts. So fortunately, they are not that clinically important.
Okay, sir. Thank you, sir.
Yes. Okay, so don't be overwhelmed. You have time, and that's why we didn't grade the exam. So I want to know how much you will retain with this rapid, fast-paced kind of online learning. That's why I really want to say I am telling you not to study for it. I'm telling you not to lose sleep over this formative exams. It's for me to see how much you retain the must knows that we are telling you during the lectures. Okay.
Okay, so wala nang questions po on my end.
you can send your email. You can send messages if there are other questions that you're uncomfortable asking me in public. Okay. So I think Dr. Baticulan wants more of your time since he didn't get much time to spend with you. Okay. Thank you so much for staying awake, but it's okay to sleep.
Thank you po.
Okay.
Thank you, sir.
Thank you.
Thank you po.
Thank you, Doc.
Thank you po.
Thank you po, Doc.
Thank you, Doc.
Thank you.
Thank you, Doc.
Ito.
Doc.